pleasant good afternoon to one and all. I am Dr.C.Kandeepan, Head and Associate Professor and the Organizing Secretary of this seminar. On behalf of the college management, teaching, non-teaching, supporting staff and students of Postgraduate and Research Department of Zoology of ARULMIGU PALANIANDAVAR College of Arts and Culture, PALANI and the THAVARAM Trust, MADURAI, I take immense pleasure to extend a hearty welcome to one and all to this one-day national webinar on cancer genomics for cancer-free world in connection with National Cancer Awareness Day. Now we are delighted to welcome our distinguished invited speaker, Dr. Abhishek Kumar. Dr. Abhishek Kumar studied M.Sc. Biotechnology from Madurai Kamaraj University and has completed his Ph.D. in Bioinformatics. He subsequently worked as a research scientist at the University of Padova, Italy, the University of Kiel, Germany, and German Cancer Research Center, Hellbach, Germany, where he developed translational bioinformatics and genomic projects. Dr. Abhishek Kumar is the head of Computational Genomic Biology Laboratory at the Institute of Bioinformatics, Bangalore. Dr. Abhishek Kumar received his PhD in Bioinformatics from the University of Bielefeld in Germany and subsequently worked as a postdoctoral fellow in the Department of Biology at the University of Padova in Italy, where he established pipelines for Roche 454 based transcriptome analysis for insect and fishes. Thereafter, he moved to the Institute of Botany at the University of Kiel in Germany, where he established multi-omic approaches for characterizing feature drug-like compounds, deduced biosynthetic gene clusters of marine fungi, and studied fungal adaptations. Currently, he has to his credit sequencing of large number of marine fungal genomes in the public domain. During this period, he established strong collaboration with several European groups from Denmark, Germany, France, and Poland. Later, he moved to the German Cancer Research Center in Heidelberg, Germany, where he worked on familial cancer genomics and developed pipelines for analysis of whole exome and whole genome sequencing data and analysis for more than 300 samples of various cancer types. He is the recipient of prestigious Ramalingaswami Fellowship awarded by Department of Biotechnology, Government of India. At Institute of Bioinformatics, he is employing multi-omics approaches for studying cancer and neurological disorders and developing computational pipelines for detailed analysis of genomes. Sir, welcome you, sir. Now we are delighted and it's a great pleasure to welcome our respected and beloved Principal in charge of ARULMIGU PALANIANDAVAR College of Arts and Culture, PALANI, Dr. P. PRABHAKAR, who is always uncompromised in his commitment, devotion and dedication in his work, and we feel lucky to have such a hardened personality to lead this institution and to show right to path as we welcome you, sir. Now I welcome to Dr. Jayakumar Raj, Chairman, the THAVARAM Trust, Madurai. Special welcomes to members of organizing committee, most respected head of the departments, faculty members, research scholars, students, scientists of various departments of our home institution and other institutions from all over India and abroad. Now I am glad indeed to welcome once again all the participants to this national webinar. We sincerely hope this webinar will greatly catalyze such interactions among research scientists, scholars, academicians, students, and pave a way for scientific and stronger, healthy India. Thank you one and all. Welcome, sir. Good afternoon, sir. Now I welcome Dr. Abhishek Kumar to deliver the plenary lecture about cancer genomics. Sir, your voice is not audible. Sir, your voice is not audible. Sir, unmute your audio.
Yes.
Yes, sir. Yes, sir. Yes, sir.
Good afternoon, and thanks for the invitation, Professor Dr. Kandeepan and Principal Sir and organizing committee. It feels like coming to Madurai, close to Madurai. Yeah, thank you very much. And let me start with the presentation. Okay, so please confirm me if you are seeing the presentation.
Okay, sir. Okay, sir.
Yeah. Are you able to see my presentation?
Yes, sir. Yes, sir. You can
Okay,
zoom in
I'm
full
start.
size. Full size, sir. Okay, sir.
Yeah,
Yeah, it's
it's
okay,
streamed,
sir.
right?
Okay, sir.
Yeah. So basically, I'm working in the Institute of Bioinformatics in Bangalore, and we are focusing on cancer. And most of the cancer biologists and cancer researchers have a dream to make a world free of cancer, right? And how cancer genomics can be helpful in this, and that's what we are going to see how what we have done and what is others are doing, and can we achieve a cancer free world in our lifetime. That should be prima facie of our lives, because it's a very complex disease, and we face a lot of challenges. And I personally get a lot of emails from others who have cancer or whose family members get cancer, and then where we land up, what should we do, and everything we are possibly can discuss, will discuss along with the research which we are doing. So first thing first, before we start, there is a wonderful website, Prevent Cancer. So treatment of cancer only comes when you have cancer or I have cancer. Why not to avoid it as much as possible from the beginning itself? So there are seven major uh, points raised by preventcancer.org, which says don't use tobacco. Yeah, tobacco, never use tobacco. So you may argue that there are some people who even do not smoke or chew tobacco get cancer. Yes, they do get it, but those who chew tobacco has much higher rate of getting cancer. Therefore, you should not live with the myth that chewing tobacco will not harm you or harm anyone, any of your family member. Chewing tobacco must become taboo. Yeah, and since the smoking cigarette industry is very heavy, we are not yet still getting stopping on smoking tobacco and other stuff. But if you make yourself aware that it's a taboo for my family, it's taboo for your family, and then it goes on, then it becomes much better. Protect your skin from the sun, eat healthy diet. That you have to do it for many other disorders like metabolic disorders. Maintain weight, be active, go for 15 minutes of walk or do exercise, practice safer sex, avoid risky behaviors, get immunized if you think that you have no immunization against HPV and hepatitis vaccines. Know your family's medical history, and it is always good to know your family's history, because from where you are going and from where you came and from where you are going is most important in every sphere of life, and so do in the history of familial history of familial history for human diseases are also very important, and it is also important for screening cancer. So any chance factor when I'm saying cancer free world, one is by treatment, other is by prevention, and prevention is much better. So on a road, if a road accident is not happening, uh, uh, it's like analogous to road accident. So if you are hurt in road accident, then you go for surgery or something. But if a road accident is not happening, then first step will be much better. So prevention is better than cure, and therefore one has to follow. Okay, I'm running a laboratory called computational genome biology, focusing on several areas. Let me introduce you to the areas where we are working. So we are working in primarily in human diseases like human cancer and other diseases, which we call omics to clinical genomics. Then we are also working eukaryotic genomes, evolutionary history of eukaryotic genomes. can we pick some drug-like compounds from microbes? And there we are also working. We have have set up in marine fungus, and we are now we have last 1 year we have initiated work on lichens, and then characterizing drug-like compounds from different fungus and bacteria. And but then when you discover drug, and when it is actually going to be effective in the for treatment, say cancer or neurological disorder, it may take up to 10 years. So therefore, again, finding a new drug is challenging. And by doing so, we are looking into the genetic diversity, the chemical diversity that exists on our planet in overall. And then when coronavirus hit, we actually started working with the viral genomes as well, starting with coronavirus. And so we're still working with this, and we want to continue in that area as well. So let me come to the what happens is, if you have a normal cell, everything goes in a controlled way. Your cell cycle is controlled, because the tumor suppressor gene which suppresses the tumor is under normal control, and the cells are growing in a normal pace or normal speed. And when there is a mutation in tumor suppressor gene, there is a no normal growth inhibiting proteins or defects in their pathway, that will lead into not so normal cell division, and that cannot be regulated. And then when the regulation is lost, you will get one will get cancer. So this is quite obvious, and these all are encoded in our genome. So our human genome has 23 chromosomes, which is about 3.4 GB in size, and with a large fraction of this genome are non-coding. So the genome, when you are talking about genome, if you're from computer science, it is a large string with character A, T, G, C. And if you are from biology, you do understand what is ATGC is: adenine, guanine, thymine, cytosine. And the random distribution of those nucleotide or bases decide how is the genome. And if there's even a single mutation can lead into diseases, and there are several studies one can do today. You can sequence entire genome, give me, and I will analyze. And then, or to an expert. An expert will first sequence the genome. An expert will then characterize computationally, and then it goes to doctor. So it's like a totally translational the report. So it can apply to other diseases as well. So the human chromosome, as I told you, 3 chromosomes are there, and the largest chromosome is chromosome 1. So if there is a critical gene in a chromosome, and in which there is a one nucleotide mutation by, say, tobacco chewing, or by sunlight or UV radiation, it can lead into a defective gene or defective gene product, which is protein, and that could lead into disease. We'll see in much more detail. And so so what today people are doing is that they are using different types of omics. Omics is like money, and genomics, transcriptomics, exomic, proteomics, metabolomic, proteogenomic, interactomics, connectomics, pharmacogenomics, phenomics, physiomics, exponomics, biblionomics, and compiling all these data with clinical data and basic biology, and then doing bio-IT, or which is called bioinformatics, to short down the data into much more meaningful data, where we can have understanding for personalized healthcare, disease, gene disposition, genetic, which will lead into understanding of genetic and molecular basis of diseases. And many of these things you can do computationally, and then requires experimental component as well. So when I'm I assume that a lot of students are there. So these are the terms here. Genomics is the getting entire genome, entire set of 3.4 GB of human genome is called genomics, human genomics, and it can be for any other disease. You can do it for insect, you can do it for fungi, you can do it for bacteria. So depending on which species you are looking for, what is your research purpose, you can today get the data by genome sequencing. Then you will get it in, maybe if a human genome, you need 4 GB of data. 4 GB of data can come in your USB stick. And now the days are not far from when you will have a watch of your own genome, and genomic kundlis will be matched to before prospective partners, and several other issues. And so we are approaching towards that. And then what is RNA-seq or transcriptomics? Transcriptomics in, suppose I am smiling and when I'm not smiling, two different conditions. What genes are expressed, what genes makes me happy and what makes me unhappy, that portion is called transcriptome. So when I'm happy, there are different set of genes are expressed means they are making protein, and therefore I make feeling happy or unhappy. So happy, unhappy is a I'm giving analogy, can be disease and non-disease condition. Then the same protein coding region is also called coding region, and when you are you found to study only protein coding region is called exome-seq or exonomics. Proteins, only protein of this organisms, all protein in a condition is a proteomics. When you are studying metabolism, it is metabolomics, metabolites actually. And then combination of all above genomics and proteomics, including RNA-seq or permutation combination, has become new area of research, where you combine all of these methods, and this is called proteogenomics. Interactomics, when their DNA is interacting with protein is interacting with metabolites, that all set of tome is the interacting partners with, so it's like a graph. If you're from computer science, it is like a graph where one is connecting with 2 or 3 nodes. And then when you are looking in the neural pathways of the brain, that is connectomics. Obviously when you apply genomics to pharmacology, it becomes genomics. Phenomics is the observed phenotypes on a genomic level. Physiognomics is again functional behavior of organism, and environmental factors is exponomics, and biblionomics is combining literature, whatever is known. Suppose say for a cancer, you first report came in 1960, and now in 50 years or 60 years later, if you want to read, want to know, you have to collect those information and appropriate way. Sometime you will not have idea or access of literature of 1960. So those things have to overcome, and that area is called bibliomics. And why it becomes so important because when the first human genome was sequenced, it was billions of dollars, and then the short sequencing, short next generation sequencing or short read sequencing came in, and then it has become way cheaper. So way cheaper, and it has reached to a level of where it is less than $1,000, which is 70,000 odd rupees for one genome. So basically, if I want to sequence my genome, I'll get it in 70,000 rupees, and then I can get an idea from it what I am facing, why I am slow or why I am fast. If I want to analyze, I can analyze. So, and it's becoming much more cheaper. So that earlier people were trying for 1,000 genome, now they are trying for $10,000 genome. Now they are trying for even cheaper, like $10 genome. And then same way the mega base pair, which is unit of a genome, a fraction of genome, is similarly is a 10 cent in dollar. So and what you see here is that this has surpassed the Moore's laws. So if you are knowing Moore's law, this is like every so I have shown you from high performance computing, 4G network, and cloud computing, that they are going in a linear way. So what was there? So in a computational term, if the Windows platform came in, say DOS, XP, slow exponentially they are growing. This is not that it's a very dramatically. So it's a linear graph normally, but now you see that this sequencing technology has actually surpassed Moore's laws. It's a technology. So much technological advancement has occurred that the cost has. Nobody expected that cost will be so low, and that you have to ask. Yeah, earlier people used, when in 2001 people were asking, oh, what should we sequence, because it's very expensive, we have to it will take several years to sequence, and therefore billions of dollars. So we have to have a very strong logic to sequence. Now today that question is gone. Today is the question, ask a research question, you can go and ask a question, you can sequence. So even in COVID-19, a lot of genomes of human which were infected by COVID-19 were sequenced recently, and it is sequenced from across globe. It sequences in New Delhi, it is sequences in Gujarat, it is sequences in Bangalore, it is sequences. I don't know how many research or clinical setup in India itself have sequenced to understand what's going in COVID-19. On similar is true for sequencing attempts of cancer across the globe. People are sequencing and trying to understand why cancer is going. And if we tap in properly, days are in I think in 1 or 2 decades we'll have much more knowledge about human diseases and cancer, that we'll be able to tap proper treatment to several type of cancer. So what it makes actually, makes in terms of data. So what happens that the as if today, so initially the data was produced on a very slow speed until late last decade, and 2010 onwards, as to approaches, the small read sequencing has rapidly started and started growing massively. Now we have 10,000 UK genome, Asian genomes, Indian genomes, several other genomes, insect genomes. So much data is available that data is rapidly growing, and every 6 months, approximately 6 months, data is doubling. So you have massive amount of data, not only from cancer and human, but from other species also. So if you just imagine that massive amount of cancer data is available in, and now it's up to us whether we can make sense out of the existing data for the future resolution of the cancer. But then we need people to be trained, adopted for such analysis. And believe me, that anybody sitting has internet and has a normal computing, say laptop or a computer, can do it from their home. And many of our trainees in COVID-19, they've successfully managed to do several analysis sitting at home. So and this data is very comparable to what is produced by major other data producing areas, like astronomy, Twitter, YouTube. And then genomic data is falling in the similar category, and parallelly it is heterologous data. I have given you what is the size of the data means. What is terabase pair? Terabase pair, probably you know. Penta is again 1,000 times more. Exa is again 1,000 times more. Zeta is again 1,000 times more. So this data is massively increasing. You need data storage. Plus, data is so much that before you plan your experiment or want to study something, you have an opportunity that initial work you can start without producing your own data, because data will be available. and that's where our laboratories is working on. And then I'm coming to the why we do sequencing, how it helps actually. So there is a recently very good article, since this, I guess, the lot of students, Jyoti and Peter Campbell has just now published a review article, and where they show that the cancer genome will provide you somatic information and germline genetic information, and then about driver mutation, chromosomal aberration, and mutational process, neo neoantigens, and then tumor heterogeneity plus pathogenesis, telomere length, and then in germline you have a drug metabolism, and as well as the familial cancer risk. And we will be covering germline in much more detail. Somatic somatic world is so massively dealt. And then here the same review is also talking about how genomics will help in cancer management actually. So basically it will help you in diagnostics and classification, pre-cancer management, and you can actually tap in genetically from a zygotic level to childhood. Screen. So basically what you can do is you can screen, I can screen my child, or a boy, or any of my relatives boy or girl, what kind of genetic markup they have, are they risk of certain cancer or diabetes or any other thing. So if I have objective clear, one can easily do it. And I think lot of genetic counseling, I think that there are officially 25 registered genetic counselor existing in the country at the moment, and number is growing. So when I'm saying 25, that means we are one of the largest at the moment in the world, and are registered with you can check it in NCBI where they have registered, and there will be much more. So the genetic counselor will tell you if you have genetic information, what kind of disease. So days are not far when you want to understand what should I do that my children will be smart, or what should I do that children my children will be sporty. You need to train then when you know the traits, and what kind of where the person can be successful, what genetic traits can be evaluated, disease traits can be evaluated. Yeah, so in a layman sense, what sequencing means, we have now human reference genome. So basically then we go into the population, take four person, one who is a very happy one, two who are silent, one who is confused. And then when they are confused in a population, you can sequence. When a person has a different type of cancer, the color coded ones, and then maybe cardiovascular one can compare them with the reference sequence and see what is the difference. That so you will discard all the commonalities and look only what is the difference with respect to reference. And also in the family, if you are looking in a family, you can see if there is a healthy one in the family, we'll go to it. So if you have a family, in the family you have a healthy individual and a sick individual. So patient, so a healthy versus patient comparison, you will do it. First you will remove everything in a family which is common to everybody in the world, and then compare what is there, what is the difference between healthy versus non-healthy individual in a family, and then tap that genetic cause for it. Can we give a drug? Can we give a personal drug to that person? So some examples I will show you. So when I'm saying that difference, so I'm trying to tell you that this is a linear genome is a linear thread or linear string, chromosomes, or if others are understanding. And then AAC is on the reference side. Now when you see that in a population there is a C, A is changing to C. At one point in cancer, the last C always change into G. So the potential cause of the cancer is that particular change of alphabet. So that, and there are databases for those population genomics is available from 1,000 genomes, GenomeAd and Cosmic, CBioportal, ICGC are the web portal for collecting data or analyzing data from the cancer. From the cancer data. So do the data for other diseases, cardiovascular, neurovascular databases are also coming in, but the cancer is most widely studied. And so what happens in this regard, that you have different cancer, you see how many genomes are sequenced in a 26 cancer by TCGA, and then when we are talking about mutation, it's like on a reference there is a T and that is changing into A, and then it can be, if it is not in coding region, it goes into regulatory genomics. If it is in the coding region, you will look what is changing at amino acid level, which is missense mutation. Okay, and that will lead us to distinguish a normal carrier versus a risk carrier, and then these will lead into pharmacogenomic for making personalized medicine. Personalized medicine, if I means when I have a disease, you will not give me paracetamol, you will check thoroughly and give me, maybe paracetamol will not work with my fever, so you'll give me a medicine which will be more effective for me rather than a generic medicine. So in the case of cancer, using omics approaches, personalized therapies are developed. So you have individual with cancer, then you have you see by RNA-seq the whole, or exome-seq, you see what is going on. Mass spectroscopy you predict the immunogenicity, and then you combine those information, and then we already know which proteins are binding against which drug, and these can be generated by a student today, by if they know the protein. Yasara, Pymol, or Rasmol are the tools where you can visualize. You can take data from mycancergenome.org, and so these are different protein data bank for the structure, and these are different inhibitors which are binding against the kinases which are actually cancer drug indeed. And so these are some examples I have given here. So what happens in this case? Suppose someone has a breast cancer, or in a population that has a breast cancer. So different individuals have different mutation. So the pink, blue, and orange individuals have a different mutation as written there. You will have a clinic sequencing, you will know them. Now you know that these have different mutation, you need to give them different drug or a different combination of drug, and therefore that's what is the personalized medicine is talking about. So it is possible for the population to know today in Bangalore or in Chennai or in Delhi by experimenting whole genome sequencing or whole exome sequencing and combining standards to know what mutations. And then there are panels also, which will be much faster, because you are not doing whole, but you are only doing sequencing of 15 or 30 cancer genes. In this case, is a HERP2 mutation is already a cancer mutation, so you are only looking for the mutation. If this mutation is present in the person is at risk of the cancer or has cancer. And so the PIK3CA has another mutation, and R has another two mutations. So these are different in a population itself. In your sample, say 100 or 1,000, you have different combination of medicine you want to give them at the end, so that they are treated, because you see they have different mutations. So they if you give them wrong medicine, it will not work. So that's when the computational biology or bio-IT is playing a role to distinguish those. And then obviously clinical screening is another. It's a work of a team. It's not an individual's work. Yeah. And so proteogenomics is playing that role, and we have developed several pipelines for proteogenomic analysis. I'm not going into detail, but what it does is that it combines data from different areas. And here we have shown one of some examples of the cancer in the right side, and the left side is generic one. And you combine mass spectroscopic data, genomic data, transcriptomic data, merge together, try to find out a meaningful result. And we'll show you one some examples. So why one need to do, because only one analysis will be not able to give you there are certain drawbacks or limitations. So DNA variants are in blue, RNA variants are in other color. Red is this somatic variant, and then you have proteomic variants are in orange. You see their numbers are varying, their sizes are telling you number, and distinguish, it's in Venn diagram, is distinguish number. So there are differences. And on the right side, these white lines are the differences. You see that there are several lines are missing, and there are white, and that shows that they in 4 different type of cancer there are different set of mutation will work, and different set of mutation will work, and therefore you need to precisely provide. So basically the first last slide to this slide is the result coming in, and these are the number of genes where these mutations are seen in a different way. So one method will have limitations, so you combine multiple method, and that's it. That is called proteogenomics, to access whether overcome the limitation of one method to other method, and to come up with a better solution for the treatment. And then when you have done those, you can actually combine on a graph to understand what kind of pathways and are those driver mutations or genes with mutations are mapped, and what kind of role they play. So there are different literature available, one can read out, and that's when you will understand fully the cancer, because cancer, as I told you, cancer is a heterologous and highly complex disease. So to understand its one method will have limitation to come up with a straightforward solution, and therefore you need to have more than one type of analysis, multiple type of data you have to produce, and multiple type of data has to be merged to form a much comprehensive result. Okay, so with this we are going to tell you what are the databases. So databases are Genomad, International Cancer Genomic Consortium, and Cosmic, plus Genomic Data Common Portal, from where you can collect the population plus cancer data set, and you can combine those and try to get the meaning if you want to just get used to it. And then what we have done, we have developed a familial cancer genomic pipeline. So these are the families in which the cancer is existing. So Hodgkin lymphoma is the family. These families are a Hodgkin lymphoma family. The red ones are Hodgkin lymphoma. And then this is what it's showing, that if you can find the mutation in the family from the if the child is getting from the parents or grandparents the mutation, there is a chance that they will get this. So you see that cancer mutation originated in second generation, came to third generation, and sometimes it comes by just marriage of a two individual which are recessive, really carrying, and then it comes in. So what we have done is that we have developed a pipeline where we analyze these methods, and using whole exome sequencing initially, and then with whole genome sequencing, which are capable of dealing with non-coding genome. And then pathway stratification is our third method, which we don't know whether we can name it as such. And so these are two papers which are recently available, and these are used widely for in our analysis. We have analyzed more than 300 families. So what it does is that we do the whole genome sequencing using it can be whole genome or whole exome, and then map it and then call the variant, then do multi-sample. By calling, you will have multi-sample family data, and also large pedigrees and trios information you can provide. Variant annotation can be performed. Annotation means you find a genetic mutation. Now you don't know what is it, so you try to match with the public databases, and these are the databases listed at the bottom. And then you have variant filtering, which you will avoid any mistakes. So you will take only high quality variant. So you will do a quality filtering, or you will do a filtering of say you will do a filtering of a population. You want less known in population, you will do the population. It should not be present in population, or it should be present less in population. And similarly, once it's done, then you will check in the pedigree where it is present, where it is not present in the family members, which family members has, which do not have. Then you do the variant ranking. That means you rank computationally that this can be a this can be a dangerous mutation, or this can be dangerous is also called deleterious or pathogenic. And then you classify where they are mapped in the genome. In the coding region, 3' region, 5' region, non-coding region, and then that are dealt separately. And then once you have it, you try to visualize it. You try to see potential relationship with the cancer predisposing genes, other resources which are already available, say human protein atlas, disease databases, germline mutation databases, and also you check the manually the sequencing read to confirm whether whatever you saw until now is not wrong. There is no sequencing error. And in the non-coding region, you are using promoter enhancer. You can read this paper. Actually, I'm briefly discussing that we check whether our mutation is located in promoter enhancer or super enhancer regions, and if so, if there is a transcription binding site, chromatin status changes, ultra conservative region, and then again go back to initial pipeline where we start checking regulatory status, and we can merge with the expression profiles. So we applied this similar mutations in a family with 3 cancer patient. One has micro PTC, thyroid cancer of different kind, and 2 of them have PTC. So we allowed that these 2 should have the mutation and not the third one. And obviously controls will not have it. So initially we get a lot of mutations, and then we end up 1970 mutations. And from 1970 mutations, we come up to a mutation in coding region 1942. And if you are understanding human genomics, you will know why. Because 98% or more than 98% human genome is non-coding. So you'll get a lot of non-coding mutations, and then you get 28 coding mutation, where we filter it coding mutation with CAD greater than 10, and then look for promoter enhancer, we find 0. And then when we do it another set of filtering, we find 4 missense variants, and these are 4 missense variants which we have considered further, and then 3 are crossed, that they are not considered, and I'll tell you next slide why it is. And then CPXM1 is the one which is carrying the mutation in this family. So we look at here, these are the name of the genes which were considered. These are the mutations or variant is called at chromosome 20 at position this, C has changed into T. And in 2 cases, obviously, then CAD score is 32, which is very high, and these all green ones are the favorable ones. So this is most favorable one, and this is located in the protein at this location. And when we look into this, we found that This mutation is in an ultra conserved region from zebrafish to human. uh, Always G is present, but in the patients we have, it become arginine or R, and that was the region, that is the region for the disease. Okay, so in a family, we pinpoint one mutation. So similarly, we applied this to several families of Hodgkin's lymphoma. And again, on Hodgkin's lymphoma, you do germline, you take information from parents, do the sequencing. The Hodgkin lymphoma is the lymphoma of lymph nodes. And so this here we have shown in the right side from Mayo Clinic, this data is taken, and the reason for the lymphoma is genomic in instability, immunodeficiency, and chronic antigen stimulation, and also lymph purification. So they all can be coupled together. So we have published several papers. Recent papers are on DICER1 as a candidate gene for familial Hodgkin lymphoma, and then subsequently other genes we have identified. So what we did was we had three families, as I showed you before, earlier, that they have, in first family have two cases, in second family we have 5 cases of Hodgkin's lymphoma. The family has other cancer as well, and the third family has two Hodgkin's lymphoma members, two children of a parents where parents had no cancer history. Get the children got the cancer. So when we start doing this as a comprehensive analysis, we pulled out all the family mutations, and then initially mutation numbers are in green, and then pedigree segregation, the mutation number comes down to very few, and then location mapping is done, exonic 3' UTR, 5' UTR in different colors, and then we find that we have identified, with greater than CAD, we identified several mutations, and then we look for cancer predisposing genes, causative HL mutations, 48 then, and finally this is the list of the top genes with the mutation in 3 Hodgkin's lymphoma family. So when we map to these data to the network or pathways, what we see that there in the first network, we see that cancer and hematological disorder, immunological disorder pathways are mutated. Second one is the developmental pathways are distorted, and third one, post RNA transcriptional modification and cell death and survival pathways are mutated. And then the peach is the color where these mutations are found. So what we see that 3 different pathways are actually enriched. And in this case, one of this case, we have identified DICER mutation. So we did the separate ranking as well. And then we see the DICER mutation, and this mutation was reconfirmed. So you take the case and control sample and try to see if you can confirm by Sanger sequencing the same mutation is existing in family or not. So we found those mutations. And this mutation is again present at the RNA nuclease domain, and what we found that there is a differential expression pattern of the microRNA that is binding to this position or to DICER1, and expression pattern, and that showed that this gene is indeed involved in Hodgkin's lymphoma. And there are also others who have identified DICER1 as the major gene for non-Hodgkin's lymphoma. So now we do a lot of cancer genomics, but maybe some of you are a student. Now I want to tell you how you can train yourself sitting at home to prepare for a research laboratory. If you want to work on such a high risk project, so you need to train, and you can train it at the laboratory, or you can be at home and train. Say you are in Palani or Madurai, or it doesn't matter where you are located, you can actually do that. So what normally we are doing is that we have a pipeline, or we can show you some of the components of the pipeline. We look for the pathway, we do the pathway analysis, we do the gene set analysis, like what is the intron size, what is the how gene is conserved on the syntenic level, what is the gene size, how is the motifs present, what is the status in population and cancer data sets. Can we find deleterious mutations and regulatory mutation? Regulatory means a non-coding mutation, and can we find them? And can we also do the mutational analysis? What we get in the database, can we pull it on the protein and see protein structural mutational analysis can we do? And that's what the pipeline is doing. And so what in one case, what we show you that these are the initial so many mutations, and then we looked into it and we found that these mutations are these ones, and then one which are yellow matching from both side are the most critical ones. In this case, the cosmic, there are green ones which are much more critical. And by doing so, we came to a conclusion that those mutations are indeed only 20 such mutations, and mostly arginine to cysteine changes are predominant, and the conservational sites and structural implications are shown here. So how we do it, that we go to, so we take this gene, we pull out our so this is a training part. You take HSP47 or any gene of your interest, look into Genomad how many mutations are available, and then we found 20 such mutations. So you can look in our paper which is published now, and see how many of these mutations are available here. Can you find them? And if you find them, what is the status? So basically, one out of the red is available here, and then sequencing quality, population density, what is the mutational rate in the population is also shown here on the right side. And so you can play around and you will try to understand, because first day, if I had no knowledge, nobody will give me this project to work with here. So you play around, take a published paper, try to see if you can identify these mutations. Not just this paper. This paper is published in Genomics, but you can take any mutation in any paper and try to look whether you can find it. Similarly, if you want to do the same mutation, you take it and you pull it in the cosmic database and see if this mutation is present, how many cases have those mutations, which cancer type has the mutation, and you can pull it out. So if you are interested, you can contact me. I can give you this paper, or you can Google it. You can get this paper, and then you try to figure out how many mutations you find from which database, and you can do repeat analysis. You can also then pull out the pathways, and correlation is in the pathways, and this is a separate paper which you can find online. It's also available free. And then you do a pathway analysis. So in best way to start is to start with one tool and then try to use different tools. So if you use a string and search, you will find a pathway, and then there are the parameters in the bottom where you say view, legend, setting, analysis, export, cluster, more, less. You play with this, see what is coming out, and discuss with your friend. Maybe discuss with so if you're confident, make it and send me. We can also play with it. And so that is the part of coding region we want to play. And if you want to play with non-coding part, it's slightly more complex. But these are two papers where the Phantom 5, and now there is a Phantom 6 also, which is explaining their mutation. So you can have a look. And then you do take a mutation. So this is the mutation which is listed there. Try to figure it out and try to see whether you can find it. So you found this mutation in the organ type, cell type, expression type. So that kind of result you'll get. It will give you a rough idea. It's not going to tell exactly about your disease condition, but it will give you a hint or promote your laboratory to do further experiment. And then when you look into the SNPA is a tool where you can find where is the mutation located, and UCSC genome browser is telling you the same position. Two different browsers I have added. So this is the mutation is in the right side, given the chromosome 13, and then what you see here, in the green or dark green or yellowish region, that there is some sort of changes happening, and then you can pinpoint those changes, the greenish region, and then you'll pinpoint those changes, that there are some regulatory nature of this variant is available. And by playing and playing, you can sometime even unravel a mystery which nobody thought before. If you do it, maybe spend some time with it. Okay, so uh, before we end, I want to give you some reading material. So you want, so are you, if you are not trained, you want to train yourself in computing, use a Linux computer, BioLinux for bioinformaticians. So the BioLinux is installed in the computer, and then you can go to the BioStars, BioTruths, and Sequencer for question answer. These are forums, and obviously other IT forums also you can use. You can use the books written by one of my good friend Prash, Your Passport to Career in Bioinformatics. Yes, you can use it. The BioStar book, the bioinformatic data skill books, practical computing for biologists, effective computing in physics, computing skills for biologists, and this first middle one is for physics, but this is general book. They have named it for somebody from physics wrote it, so they say physics, but it is valid for everyone. And most of the books, it's not nothing to do with biology much, but obviously biological examples will be there. But in life, every example is can be correlated with other examples. Okay, so what kind of team we have? We have a very strong team working on a various aspect of the project. So there are several smiley faces, and some of them are master students, some of them are Ph.D, some of them are training, and some of them are postdocs. And then during COVID, we have several team members who work on as a COVID volunteer. So anybody who want to join the team can ask us via email. And also, if you have a query, you can reach us. And if you are looking for a job also, you can reach us on India Biosciences, GitHub, Job Archive, ResearchGate, everywhere our advertisements are available. And also you can go to our website. Yeah, so some of the smiley faces, we are looking forward to meet them through this talk when you reach us and you have done. So this year we have in pandemic, there are 10 master or bachelor students have did their thesis actually. So we are thankful to several funding agencies, Institute of Bioinformatics, ICMR, Department of Biotechnology, and BioBAM, Omics Box, and Manipal, where I am professor or associated faculty. Yeah, so with this, I ended my talk. And so I'm open for the questions. So I can also take, go to the question in live chat in the chat, and you can also raise some of the questions. So I'm open to answer the questions.
Sir, I have question, sir.
Yeah.
Okay, sir.
So we have some question in WhatsApp, right?
Yes, sir. I posted the questions when when the participants are registered. They are raising a lot of questions. This one is Salina Subhas. What is the genome testing for cancer?
Genome testing, what do you mean by genome testing? So basically, you have to do the genome test. So that's what we were talking about all the time. So what happens in the genome? The normal genome will have no such mutations, while cancer genome will have the mutation. The mutation is missing, hence it will be available in cancer. And sometimes it could be large mutation or large insertion. So some cancer is happening because of viruses, right? So like HPV. So HPV will change, has to integrate in your genome, right? So that kind of test one can do, and genetic testing is quite common, and that's what we're talking when we're talking about mutations in the family. Yeah,
So
please, next question.
next question, sir. Why does the cancer reoccurs even after the tumor cells are removed by surgery?
Yeah, so life is a recurring process, right? So tumor is removed, but suppose the genetic markup. Sometimes person gets so in the family also, family members have different cancer, or even a single member has multiple cancer. Okay, so what happens if you have a genetic markup? So your cancer will reoccur. Your tumor is removed, but the genetic marker is same, so it can reoccur after 5 7 years. So one of the person I last met last year, he has 10 years back cancer X, and then after he worked 10 years, he moved in life, living happily, and then he got cancer. Why? And he said, how come it can happen to me? See, it's your history, genetic history, and also what mutation you have acquired during your life has the role. So one has, so if you know that water in some area is contaminated, you must take care of it immediately, because that will lead into some mutation which may occur. And again, for the same tumor, it can happen. No, when you have this history or with the mutation, the doctor try to take as much as possible. Sometimes a minor tissue is left out, because this entire organ they cannot remove. Your entire tissues of certain areas cannot be removed. It's not like cutting potatoes. It's your organ where you have to survive after the surgery as well. So doctor will take utmost care, but then they also will have to deal with the situation. And and as again, if it is in your its mutation is existing in your genome, there are chances again. And one of my close friends sister recently got one cancer, and I recommended her once she was recovered from it. So I recommended her, and every now and then we are counseling that we should go for eating as healthy diet as possible to keep herself healthy in the future, and not let the reoccurrence happening. So there are several different regions for the cancer. So another question: how to how to select cancer cell line while studying molecular docking. So what happens in the molecular docking? You know what cancer, what drug you are looking for. So if the cell line has, if the drug has an effect on the cell line, then you can choose, but you need to have extensive literature available. What is the symptoms for early cancer? Symptoms for early detection of cancer. Symptoms for early detection of. This question is confusing. So you want to detect early. So you meet acute genetic counseling as early as possible. And now many hospitals, Convenor hospital, I think in Delhi, and other hospitals also in Bangalore, they are doing the screening for the child as early as they are born. So to identify if they have some disorder. So and childhood cancer is also very common. So therefore one need to be very careful. The environment, the food they eat, the care they should provide to their children.
So, so what are the symptoms
Someone
for
is
early
asking what is
detection of
the
cancer?
procedure of being part of my team. You write a good application, send your CV to me, and if we have requirements, and also if you like your CV, we will discuss with you for sure. So Gayatri has asked how to avoid breast cancer after 35. Yes, breast cancer is getting very common, and also it's very, so one has to be as aware as possible. If the if you have any other abnormality in your breast, or you normally should also meet a doctor for checkup. We should not shy away with our own organs. And also, we should not shy away with any situation we are in. It needed to be openly discussed. Suppose I am getting COVID right now, then I am hiding. How long I can hide? So similarly, I request all of you to not to shy away. Let's bring this social stigma out that we can't discuss, and so and educate others, not just for yourself. Educate others. Lot of women in villages are not able to talk. Uh, they think uh, taboo to talk about uh, their organs, but it's not. Should not be like that. It is must that if you are facing pain, if you are facing bulge or swelling, you should meet a doctor and take as early as you detect the symptoms. Chances that you will be saved is much better. And so for any just not for breast cancer, for any disease, you one should not shy away, and one should look for the solution, meet doctor as soon as possible. And also, when people are aware, they will do more and more.
Sir, one more question. At the age of 42-year person suffering from blood cancer, or we all, what is the recovery percentage?
Again, that doctor will tell in what kind of what stage the person was identified. It is a rare cancer. It depends. It depends where you are treated, where what kind of stage you are treated, and very rapidly treatment regimes are growing up. So basically, one need to meet a doctor immediately. Will there will be a complete remedy for cancer in future? Complete remedy. Cancer is not one disease. There are different type of breast cancer, and so on. There are different kind of organs which cancer occurs, and for even for one type of cancer, there can be multiple regions. So uh, personalizedly it has to be tackled. One remedy for one stop solution, no, one stop solution will be not be there. One stop advices one can give there. Okay, don't see tobacco as I said in the beginning, and do be healthy. And these are not just for cancer; these are for living healthy. Right? So any other disease as well. How
Uh,
does oncology in our body contribute to cancer growth? Obviously, the definition of oncogene is that gene potentially has a mutation, and it will regulate the uh, pathway pathway in pathway context, or suppose gene is producing a protein and its active site is mutated. I can give an example. Uh, Suppose this is the protein and it has to bind here, but it has a mutation. Now, due to this mutation, the binding it is not able to bind, or able to bind, but with not same efficiency. It will lead into problem. So, oncogene mutation in oncogene will lead into disease in a similar way. And then, can you get a clear picture of human cancer by studying genomics and proteomics? Does epigenomics play has a role? Yes, cancer genomics, proteomics, epigenomics are technologies or methods, and you will get a lot of ideas from them. Epigenetics also play a role, and then again, I'm telling you, this is a complex process, so you have to apply multiple methods, multiple approaches, and you need to combine a lot of literature and a lot of a lot of information uh, from different sources. And then this question: what is the nanotechnology and biotechnology? Obviously, you can provide biotechnology and nanotechnology also. Basically, if you are a biotechnologist, actually you are studying biology in prima facie, and if you are grow your knowledge, you will be playing role. Nanotechnology definitely. has play maybe in a drug discovery capsule, and several areas you can read about. Nanotechnology is coming very rapidly. Is there any particular screening cancer? In silico method for screening cancer. Obviously, you can, if you have a genetic data available, you can screen entire rest of the things in from NGS data. By and large, computationally, but it's not a straightforward process. Is there any so what is okay? This is done. Yeah. So there we have answered a lot of questions, and I hope that helps. And if those who have certain questions can reach me, uh, reach me. Uh, Annapurna, I have seen your question, but I have to read it. Yeah. So before that, any scientist should not answer half-heartedly. So you can reach me. I will answer you. But before that, I have to read your question. Read about your question. Okay. And yeah, so keep on asking question. Maybe I will be on the chat later on and answer some of your questions. So I will really thanks organizers. Sir. Tanu Das is asking that cancer spread from patient to healthy. No, no, it is not infectious disease. It is when I'm saying it is found in family. It is coming genetically. So genetically, or the same family is living the same environmental condition. So if the parents are heavy smoker, the children are getting uh, indirect smoke and living the same condition, and so they can also get the same similar cancer, but it's not infection. Taking oral chemotherapy for years together is safe. I'm a researcher, so basically, oral chemotherapy, it has to be given under the precise guide of a cancer doctor or chemotherapist, and so and safe or unsafe, it's totally the doctor will tell. And I'm obviously chemotherapy is not the safest method, but that for sure. And you see a lot of symptoms post recovery, but people take it when they have no other choice left. So therefore, I would not recommend you to take it from a shop and start doing it. You should consult doctor, and every not for years. You should take it consult every 3 months so, to doctor. Okay, with this we are thankful to all of you. Those were there. Thank you. Thank you, sir.
Thank you, sir. Thank you, sir. Now I welcome Dr. Jayakumar Raj from the THAVARAM Trust Madurai to deliver the vote of thanks.
Sir, thank you, sir. At the outset, let me thank the management, APA College of Arts and Culture, PALANI, for joining with us in organizing this one-day national webinar on cancer genomics for cancer-free world. I take this opportunity to thank the management for their support and encouragement in organizing this webinar. I personally take this opportunity to thank the principal, APA College, for readily accepting the invitation to organize this event and supporting us throughout in organizing this webinar. I express my gratitude to Dr. S. Ramya, Director, THAVARAM Trust, Madurai, for supporting us. I express my deep sense of gratitude to the speaker, Dr. Abhishek Kumar, Faculty Research Scientist and DBT Ramalingaswami Fellow, IOB Bangalore, for having delivered such a wonderful speech on cancer genomics for cancer-free world. Sir, you have enlightened us in this field with very simple words so that everyone can understand that everyone includes the students, the research scholars, faculty members, and all other participants who have directly involved themselves in this process. You have enlightened the young mind to participate and take up research career in this field. I thank you once again, sir. I thank the organizing secretary, Dr. C. Kandeepan, for his efforts in organizing this event in a successful manner. Thank you, sir. Let me take this opportunity to thank all the participants who have registered and participated in today's event. I hope that this event would have definitely thrown knowledge on this very field of cancer, where they wouldn't have listened to such kind of things in a simple words and got a clear understanding. So this is definitely going to enlighten everyone in this field. Thank you once again, one and all, for participating in this field. Thank you.
Thank you, sir. So thank you, sir. Thank you, Abhishek Kumar, sir. Dr. Abhishek Kumar, you delivered an excellent speech for us. Thank you once again. Thank you, sir. Thank you.
Thank you. Have a nice
Thank
time.
you, sir. Thank you, sir.
Thank you, sir. Thank you.
Thank you, Dr. Jayakumar Raj, for your
Thank you.
deliver
Thank you,
the
sir.
vote of
Thank
thanks.
you.
Thanks. Attention to the participants. The feedback link will be posted in below the live video in description. So you have to fill the feedback link and send it. Definitely you can receive the certificate within 2 or 3 days, because Google creates only 100 certificates per day. Thank you one and all. Thank you.